Good afternoon, everybody. In the following up on last Wednesday's lecture on XMRV, remember that? Anybody listen to it yet? I got an email from a patient. I used to get lots of emails from CFS patients, and this just came in. Hello, Professor. A new gamma retrovirus article in the vein of XMRV was just published. I'm hoping to get your take on it. The abstract says, unlike earlier reports in which MLV-like sequences were identified in human source material, which may have been due to murine contamination. May have been due. What the hell is the may doing in there? It was due, right? Sorry, continuing in the abstract. Budding retrovirions were demonstrated repeatedly by electron microscopy in uncultivated lymphocytes of the patient. And immunological evidence was obtained that the patient produced IgG antibodies that bound to the particles. These data indicate that the patient had been infected by this virus, which they're calling JHKV, and lending significance to the demonstration of amplicons and nucleic acids from the patient's PBMCs. So we'll have to check that out, but as you can see, I see the author. I see one of the authors here. So I'll bet this is contamination too. But we'll have to see. Just be skeptical. So don't believe any one thing. As you learned last time, it's very easy to believe that there is a virus causing your illness, but it does you no good if it's not correct. Now, what I want to do today is to tell you another story of viruses in the news, but this one is really different. This one really affects our ability to do research in the future. And for those of you thinking about doing research, you should know this story and its implications. And it's called H5N1. And this is an influenza virus. Remember these viruses? Here's a beautiful image of an influenza virus, which should by now look like a sibling to you. You should look at this and say, yes, that's influenza virus. And this is a particularly beautiful image, three-dimensional image, which shows you the viral membrane and even the M protein shell beneath it, that purple thin layer there, the hemagglutinin and the neuraminidase on the surface. And look, the M protein ion channel. Isn't that great? You can see it goes right through. This influenza virus we're going to talk about today is one of many different kinds of influenza viruses, as we've talked about in this course. And as far as we know, out there are influenza viruses that have 17 different HA hemagglutinin types, right? The blue protein is the hemagglutinin. So we know of 17 different kinds of HA, 9 different neuraminases. And these are all out there in various animals, as you'll see in a moment. But only certain types are human viruses. The H1, H2, H3, the N1, and the N2. Those are the only ones that transmit from person to person, repeatedly cause epidemics every year, and pandemics every now and then. So avian influenza is a different disease. Birds are the natural reservoir of every influenza A virus that we know about. All those HAs and NAs are in all kinds of birds, particularly in these water birds, ducks, geese, swans, gulls, terns, and shorebirds. All right, there are many other kinds of birds as well. They're in poultry. It's bad when they get in poultry, of course. So this is where we think all the influenza viruses came from, and even the ones that infect us probably originated in birds. In birds, the infection is very different from in people. It's a gastrointestinal infection in birds. It replicates in the GI epithelium. It's shed in the feces. And this car is covered with bird feces. I guess it's been parked under a tree for a while. It's not my car, by the way. And that feces can be full of influenza viruses. So it's very different from human flu, which you cough and sneeze and expel it. Birds defecate, and that's how they release their influenza viruses. So as birds are flying overhead, they're releasing influenza viruses. When they come down to the ground and feed in the water, or swim in the water, they're releasing influenza viruses into the environment. So there are very good mechanisms for spreading the virus. The virus does replicate in the respiratory tract, but to a much lesser extent than it does in us. And in many, many birds, many of these water birds, the infection is typically asymptomatic. There are some exceptions in wild birds, but for the most part, avian influenza infections in wild birds are asymptomatic. In poultry, they are not. They often make poultry very sick or kill them. Now, avian influenza viruses are classified according to what they do in chickens, which of course is a bird that we raise for eggs and for food. And there are two kinds. There are what are called low pathogenicity avian influenza viruses, LPAIs, and these cause a mild respiratory disease in birds, and they decrease the egg production, so you don't want to have these in your flocks. And many chicken farmers immunize their birds against various influenza viruses to prevent this. Then there are high pathogenicity influenza viruses, and these are typically of the H5 and the H7 HA types, subtypes. These are typically lethal in poultry, in chickens in this particular case, which is what we're defining, basing the definition on. And as I said, in wild birds, they're usually not. One of the defining features of these highly pathogenic influenza viruses in birds is a basic amino acid sequence at the HA cleavage site. Now, you may remember from a long time ago, we talked about how the HA has to be cleaved in order for the virus to be infectious. And the cleavage occurs right here with the arrow to release the N-terminus, which contains the fusion peptide. So cleavage is needed for infectivity. And most influenza virus strains have a sequence at the cleavage site that is only cleaved by proteases that are present in the respiratory tract, and that's what limits these viruses, that's what limits their tropism to that organ. But high pathogenicity avian influenza viruses have a basic amino acid sequence here, which means that it can be cleaved by ubiquitous proteases that are present in every tissue in our body and in birds' bodies. So the virus can, in fact, spread beyond the gut tract in birds or the respiratory tract in humans. So this gives them a lot of their high pathogenicity. There have been many outbreaks of high pathogenicity avian influenza viruses, probably always, but since we've been looking for them, and this slide is since 1955, and you can see the name of the virus here and the subtype. And again, these are typically H5s and H7s. And all the birds that either died or had to be killed. So look at this one, is a big one in Pennsylvania in 1983. 17 million birds either were killed by the virus or had to be culled. So when you have outbreaks of these viruses on farms, chicken farms or turkey farms, goose farms, you have to kill all your birds to prevent the spread of the virus. And so that often is a lot of birds, millions and millions of birds on some of these farms. The boldface fonts are the ones with over 1 million birds that that either died or were slaughtered. Millions, millions, millions. And these continue to this day. There are outbreaks up until 2013. I just haven't listed them here because it's more of the same, essentially. Now, in birds, there was an outbreak of influenza, H5N1, a high pathogenicity version in the live bird markets of Hong Kong in 1997. So this is where this story begins. As many of you may know, in various parts of China, there are live bird markets where you can go and purchase birds of various sorts and have them slaughtered, and then you can take them home and cook them. So there's a lot of opportunity for contact with raw meat, and you can get infected with avian influenza viruses in these markets. This virus that emerged in the bird markets in 1997 was a reassortant of various avian influenza viruses that went to produce it. And along with this outbreak in 97 were the first human infections with avian influenza virus. So it had never been seen before, which is not to say that it never happened. We just never noticed it. This was the first time that we picked up human infections with avian influenza. There were 18 people confirmed to be infected, and 6 of these died. And as a consequence of this outbreak, all the poultry in the markets were slaughtered, and this stopped the spread of the virus, but it was already out there in the wild most likely, and it reappeared in poultry and wild birds in 2001 and 2002. There was subsequently an outbreak of high pathogenicity H5N1 in July 2003 in Vietnam, Indonesia, and Thailand. That virus, a slightly different virus from the 97 one, spread to over 60 Asian, European, Middle Eastern, and African countries. This virus is now enzootic, which means it's there all the time in poultry in China, Indonesia, Vietnam, Egypt, India, and Bangladesh. So it periodically causes outbreaks and periodically leads to human infections. This virus was spread globally by migratory birds. It spread very quickly. Within 10 years, it had gone to most parts of the globe. Humans also helped to spread this H5N1 virus by transporting infected birds. There's a very large illegal bird trade globally, and some of that spread it. And in Thailand, the zookeepers fed infected chickens to the cats in the zoos, the lions and the tigers and so forth, and they got infected because the chickens turned out to be infected, and the people who were feeding them to the cats didn't know that. So the virus can not only infect birds and humans, but also other kinds of mammals. So in people, high pathogenicity H5N1 infections are very atypical for influenza. There's a very aggressive clinical course, which means that the infection proceeds rapidly. There's often lower respiratory tract involvement all the way down into the alveoli, and the whole syndrome is called acute respiratory distress syndrome. It's a very serious disease and often leads to death. It's often accompanied by gastrointestinal symptoms, diarrhea, vomiting, abdominal pain, which are typically not present during infection with most seasonal human influenza virus strains. There is no transmission of these viruses among humans. Right? This is something we will return to very shortly. All these people who are infected by avian H5N1 virus appear to get it from a bird source of some kind, the live bird market or some other contact people who raise birds in the countryside, for example. In many countries, the birds are in the same room as the people, and it's very easy to acquire infection. And people at risk are those who work with birds, those who kill them, defeather them, handle them, prepare the poultry, all kinds of levels of involvement from the farm all the way to the meat market. So this virus, H5N1, is considered to have pandemic potential. And that means it has the potential to cause a global outbreak. Very few people have immunity to the H5 hemagglutinin because very few people have seen it. And it's in poultry. It's endemic in poultry. It's all over the place. Well, not in every continent, apparently, but it's in many places. So it has pandemic potential. Here's the geographic distribution of this virus globally. The light colors show you H5N1 in wild birds, which are in a number of places, and WHO and other agencies do a lot of global surveillance to see where this virus is because people are very worried about it. In poultry and wild birds are in these darker colored countries. You can see quite a widespread distribution, and then the red squares are where the H5N1 human infections have occurred. And you can see it's not in North and South America. And this is probably because the birds have not brought it there. The migratory pathways don't allow the birds to transmit it from other parts of the world, and luckily humans haven't brought the virus in either. And these are the cumulative number of confirmed human cases of infection that have been reported to WHO. WHO has a very strict case definition for H5N1 human infection, which we'll go into in a moment. And here's the total on the right here. You can see the countries that are involved. And you can see some have a greater burden than others. Indonesia and Egypt, a lot of cases, and Vietnam. Those are the three countries where it's the biggest problem. 628 confirmed cases and 374 deaths. And this table is updated periodically, and you can find it there. So the case definition by WHO is very specific. It's an acute febrile respiratory illness, so that's influenza so far, with known exposure to an H5 virus in the preceding 7 days. So there has to be some kind of exposure in a poultry market or on a farm. You have to have molecular confirmation of infection by a WHO lab. Can't be a non-WHO lab. It has to be WHO, so this could be PCR, for example. And this rules out asymptomatic infection. So if you are asymptomatically infected by H5N1 virus, you won't be recorded by WHO because you have to have an illness. So this case definition is very strict, and what it does is excludes rural areas with poor health care, where in fact most H5 cases occur, because you're not going to be able to get to a WHO lab to confirm the case. And if there are asymptomatic infections, you're simply going to miss them. So there are lots of missing numbers in the equation to figure out how lethal this virus is. But if we just take the number of WHO confirmed cases, that's 628, and then we take the number of deaths among those confirmed cases, we get the case fatality ratio of 60%. All right, so case fatality ratio is number of deaths divided by the number of confirmed cases. This is the fear factor for H5N1. That's a pretty high number. And many people think, wow, if this virus began to transmit from human to human, 60% fatality would be really high. It turns out that the mortality rate, which is a different number, is probably much lower. Mortality rate is the number of deaths divided by all the infections, not just the ones that are confirmed by WHO and which have overt clinical symptoms, but all the cases that occur. We just don't have that number at the moment. We don't know how many H5 infections actually occur. There have been some studies, but they're all inadequate for one reason or another, and this is a question that really needs to be addressed. Here's one paper published last year from here in New York City. Seroevidence for H5N1 infection. This is actually a meta-analysis. They looked at the literature and took out all the papers where people looked for antibodies in people against H5N1 virus. And they concluded that 1 to 2% of more than 12,500 study participants from 20 separate studies had seroevidence for prior H5N1 infection. All right, so this is very important if this is true. We don't know if it's true. We don't know if these are cross-reacting antibodies. We don't know how substantial the antibody levels are. There's a lot of questions, but the possibility remains is that there are a lot of people out there, especially in areas where people have contact with birds that are potentially infected. Vietnam, for example, where the virus is known to be endemic. So just keep that in mind as we continue this discussion. Just for comparison, let's look at the numbers for a seasonal human influenza virus infection. So this is the 2009 H5N1 strain. This emerged in that year. It was a pandemic strain, spread globally. And these are US numbers. There were, from the CDC, 12,469 deaths in the US, 274,000 hospitalizations. So if you take those two numbers, divide them, you get a case fatality ratio of 4.5%. So these hospitalizations, these people are sick, they have symptoms. It's not an inapparent or mild infection. So that's the case fatality ratio. But CDC has estimated that there are 60 million cases. And if you then take 12,000 deaths and divide it by 60 million, you have a lethality or mortality ratio of 0.02%, which is more in the line with seasonal influenza. This turned out to be a particularly mild pandemic strain. So you can see that we have 628 deaths from H5N1. That obviously in itself, while those people would rather be living, that number is far lower than the 12,000 that we get in a pandemic year. There's another study that I want to tell you about that was done, which measures a different parameter, and that is it measures whether patients contain T lymphocytes that can recognize the H5N1 virus. I'm really sorry about this feedback, and when I try and reduce it, it puts my voice out entirely. So in this study, you remember earlier in the course we talked of how T cells can recognize virus-infected cells via the T cell receptor. So if there's a virus-infected cell displaying viral antigens on the surface, the T cell that is specific for that antigen will recognize it, and the T cell receptor will bind the antigen, and then the T cell will kill the infected cell. So these are cytotoxic T lymphocytes. So what they did in this study was to make peptides, short peptides that would fit into this T cell receptor, which represented all the possible peptides from an H5N1 virus. So then they would take lymphocytes from patient blood and purify the cells out, and then add peptides and ask, do these peptides, are they recognized by the T cells? And there are ways to measure that. The T cells respond by making various cytokines. They found that 20% of the patients they looked at, this was a high-risk Vietnamese cohort. High risk because these are people near poultry farms, and we know that H5N1 virus is present there. 20% of them responded to H5 peptides. That means they either had a subclinical H5N1 infection and survived, obviously. If they had a serious infection, we would know that. Or maybe there was some cross-reactivity with another virus. We can't rule that out. So many people don't believe the serology that I just told you about, that 1% to 2% of certain populations are seropositive for H5 antibodies. But this is a totally different test, and I find that this is compelling. This suggests that there are people out there that have been infected that aren't getting sick. Now, if you have looked in the news at all in the past few years, you know that H5N1 is a big deal. And the press always touts it as an extremely lethal virus based on the 60% number, the case fatality ratio. But I contend that that's not correct. Until we know what the actual mortality ratio is, we should restrain from making dire predictions. So how much should we really worry about this virus? Well, it's endemic in poultry. There's this case fatality rate issue, which I think has to be resolved. It doesn't transmit among people. Will it ever? I don't know. It's been circulating a long time. It's had a lot of chances. Maybe it hasn't had the right encounter. Maybe the right virus in an animal hasn't found the right human host. Maybe that could happen. I don't think we can really predict. I don't even think you could give a number on whether it would happen or not. The only thing I can tell you is that only H1, H2, and H3 viruses have transmitted among humans for as long as we've been studying this disease. So I don't think we can predict if there will be an H5 pandemic. And that really is important because we spend a lot of money on this virus surveillance and preparedness. We have vaccines and antivirals. This is a very interesting question, which I don't have an answer for, but it's clearly important. There are other avian influenza viruses that infect people, and they don't get much attention in the press. For example, in 1998-99, H5N1 viruses were transmitted from birds to both pigs and humans. These were not fatal infections, but they were confirmed influenza. Interesting outbreak of H7N1 in the Netherlands in 2003. There were something like 250 different poultry farms affected by this virus, 89 confirmed human infections. There was some human-to-human transmission, and there was one human death in this outbreak. In 2004, two humans were infected with H7N1 after an outbreak in poultry in Canada. So other avian influenza viruses have the capacity to infect people. They don't get much attention because they don't have the 60% case fatality rate, and the numbers are much lower. But these could be as much of a threat. Now let's talk about the receptor for these viruses, because this becomes an important issue in our discussion. You remember we did talk a long time ago about how influenza viruses bind to sialic acid containing glycoproteins on the cell. That is the receptor. And avian and human influenza virus strains prefer different kinds of sialic acid linkages. So avian strains prefer alpha-2,3 linked sialic acids, which is shown here. So remember, here's the glycoprotein. That's the protein part in blue. These are the sugar residues, and the terminal sugar is typically sialic acid. When sialic acid is present, it's the terminal residue. And the virus binds to that. This is an alpha-2,3 linked sialic acid. It means the sialic acid is linked to the next sugar by an alpha-2,3 linkage. That's preferred by avian strains. Human strains prefer alpha-2,6 linked. So the alpha-2 to 6, this carbon up here, that would be linked in the receptor that human strains prefer. All right, that's an important point to keep in mind. In our respiratory tract, these two different kinds of sialic acids are present, alpha-2,3 and alpha-2,6. We have both, but the distribution is different. So if you just look superficially, the nasal mucosa, the paranasal sinuses, the pharynx, the larynx, these are sites where the virus would first come into you. They have mainly alpha-2,6 linked sialic acids. Then as you move lower into the trachea, the alveoli, the bronchi, the bronchioles, now we see a mixture of alpha-2,6 and or alpha-2,3. So alpha-2,3 is present lower down. And the conventional wisdom has always been that to acquire an avian H5N1 infection, you had to inhale very deeply a large dose of virus so that it got at least into the trachea, which is not trivial because there's a lot of filtration going on before that. However, if you look in the very upper tract, you can see there are some alpha-2 linked sialic acids in the cornea, in the conjunctiva, and also in the nasolacrimal duct. So it's very possible that you could be infected at, say, the cornea or the conjunctiva. Virus could replicate, go down the nasolacrimal duct into the respiratory tract, and then move down. So you don't have to inhale a whole lot of virus in order to do that. And in that Dutch outbreak, I told you about the H5N1 outbreak, many of those people had conjunctivitis initially. So this shows that those viruses can infect the eye of people and perhaps get into the lung by these routes. Now the real question is, why doesn't H5N1 virus transmit among humans? So we know this is the case. Those 628 infections are always bird source to person, not person to person. Of course, you need an animal model to study this. You can't do any kinds of H5 infections in people. It's a lethal virus, and that would be, of course, unethical. And one of the models that's been used is the ferret, which is this guy here. And ferrets have respiratory tracts that are very much like human tracts. They can be infected intranasally by a variety of influenza viruses. They develop a sneeze reflex, so they make an aerosol, and they can transmit viruses to other ferrets. They get runny noses. They get influenza. They get a fever. They make an immune response. So this is a model for looking at influenza virus infection and transmission. There is another model for transmission that we've talked about, guinea pig. And these guinea pigs can be used to show virus spread from one animal to another. But guinea pigs don't display all these symptoms of influenza like ferrets do. So they're the poor man's model for transmission. They're much cheaper than ferrets, but guinea pigs don't sneeze and cough, and all the transmission that you're looking at is just from the exhalation of viral aerosols from the guinea pig. In this model, people have studied H5N1 transmission. And one of the things that's important to know is that just changing the sialic acid specificity of H5N1 isn't enough to get the virus to transmit from ferret to ferret. So if you take H5N1 and you make it recognize alpha-2,6 linked sialic acids, which is pretty easy to do, the virus will replicate in ferrets, but it will not transmit by aerosol. So you need more than that. In addition, if you make changes in the hemagglutinin, the neuraminidase has to match it. The affinity of the HA for its receptor has to be matched in turn by the ability of Na to cleave off the sialic acid. There are also changes in the RNA polymerase that are important for getting avian strains to replicate in mammals. In particular, one of the RNA polymerase subunits, there's a very specific mutation, which we'll encounter in a moment, which allows the avian viruses to replicate at the higher temperatures of mammals. We have higher temperatures than birds, so their viruses don't replicate very well in us as a consequence unless you put this amino acid change into the RNA polymerase. So the real story begins not just with H5N1, but with these two virologists who I've known for many years. This is Ron Fouchier on the right, he's in Rotterdam at the Erasmus Medical Center. And this is Yoshi Kawaoka, who is at the University of Wisconsin-Madison. And they both work on influenza virus. And they're very interested in what regulates H5N1 transmission among mammals. So this is an important question. We'd like to know what controls it so we could see if a strain were emerging that had the potential to transmit among humans. So they did a series of experiments which basically lit a fire on the virology community, alerted the world, alerted governments, and now, as a consequence, we are all paying the price for this. And that's what I want to tell you about today. It all began with this article in Science Insider by Martin Enserink, which was published in November of 2011. Scientists brace for media storm around controversial flu studies. The first sentence, locked in the bowels of the medical facility building here and accessible to only a handful of scientists, lies a man-made flu virus that could change world history if it were ever set free. Now, I have told Enserink that these are among the most inflammatory words that he could have written, all right? And he said, well, that's what Fouchier told me. So I just wrote what he told me. This was based on an interview with Fouchier after he had done the work, and which we'll talk about in a moment. The virus is an H5N1 strain that has been genetically altered and now is easily transmissible between ferrets. Scientists believe it's likely that the pathogen, if it emerged in nature or were released, would trigger an influenza pandemic, quite possibly with many millions of deaths. This is another bit of an overstatement. And Ron Fouchier says his team created probably one of the most dangerous viruses you can make, which was not a very smart thing to say. And this triggered an intense debate about the limits of scientific freedom. And then there's another study quoted here done by Kawaoka, which basically did the same thing. Yoshi didn't respond to interview requests. I think it's probably a good idea. He probably should have said something, but not what Fouchier said. Now, these papers were submitted for publication towards the end of 2011, and they were intercepted by a board in the US called the National Science Advisory Board for Biosecurity, the NSABB. And this is a board that was formed after the anthrax attacks in the US. Remember, after 9/11, a few envelopes with anthrax spores were sent to people, and a number of people died. So the US says we need a biosecurity team, and this was put together. And what they do is if a paper is submitted to a journal that looks dicey, it could be used for bioterror. This committee kicks in and they look at the work and they could decide whether it should be published or not. So these are two members of the NSABB at the time. Paul Keim, who was an anthrax person. I can't think of another pathogenic organism that is as scary as this one. I don't think anthrax is scary at all compared to this. Okay, if you think so, but as you'll see by the end, I think this is mostly an uninformed opinion. And then Richard Ebright, who is a molecular biologist at Rutgers, who studies RNA polymerase II. This work should never have been done. And these are sentences you should never say as a scientist. I think you have to be really careful, and I want you to form your own opinions by the end of this. So what did Fouchier do? Turns out that in September of 2011, he presented all these results at a meeting in Malta. And there were lots of flu virologists there. And what he said is he had introduced 3 changes into the H5N1 to adapt it to mammals. And we guess that these are changes to adapt it to alpha-2,6-sialic acids and a change in the polymerase to let the virus replicate at the higher temperatures of mammals. This virus did not transmit among ferrets by aerosol. Then he passed the virus from ferret to ferret 10 times. So you take a ferret, you pipette a little virus into the nose, you put it back in its cage, you let it incubate for a certain amount of time. Then you sack the ferret, you grind up the respiratory tissues, you get the virus out, and then you infect another ferret. So that's a ferret-to-ferret pass. 10 times. He called it a really stupid project, and people were wondering, why did he call it stupid if he wanted to do this experiment? It turns out apparently the translation in Dutch is an obvious, he meant to say an obvious experiment, and he got the translation wrong. Or so it goes. And finally, this virus that he passed could then transmit in the air among ferrets. So people who went to this meeting knew what all the changes were, and none of them would tell me. I would meet them at other places, say, what are the changes? And they wouldn't tell me. This is an article that came out in New Scientist 5 easy mutations to make bird flu a lethal pandemic. Where does this come from? Where's the data that this could make a lethal pandemic? This is totally a huge jump. Anyway, here it tells you, again, what I've just told you, the 5 mutations in 2 genes that allow the virus to spread between mammals. But look at this sentence. This is the key. The virus is just as lethal despite the mutations. Fouchier said it's transmitted as efficiently as seasonal flu. Peter Doherty, a Nobel Prize winner, he didn't read this paper, by the way. He said, this shows clearly that H5 can change in a way that allows transmission and still cause severe disease in humans. It's scary. Did you know that ferrets were humans? I thought this experiment showed that it could transmit among ferrets, and we'll come back to this just as lethal suggestion. Okay, so you can read the rest yourself. Let's go to the NSABB and talk about their work a little. So this is an advisory committee to provide advice, guidance, and leadership regarding biosecurity oversight of what is called dual-use research. This is a new word in our lexicon that has appeared post 9/11. This is research with legitimate scientific purposes, but you could misuse the results. You could make a weapon of some kind, and it could have a threat to public health or national security. So that's why this committee exists, to look at potential dual-use research. So there's a whole list of things that fall under DIRC, as it's called, and they would examine this. So this is staffed by a variety of people. You saw Keim and Ebright are on the NSABB. These are various experts in different fields, not just science, but law, publishing, biodefense, national security. Can you imagine getting in this room and talking about sialic acid receptors with a defense person who just wants to blow up the world, right? So you have a real mix of interesting people here. Now, when you submit an article to a journal, and I know the American Society for Microbiology journals, there's a little checkbox that says, check this box if these experiments are of dual-use concern, I think is the wording. And if you review the article, you have to check that as well. And we interviewed one of these NSABB members on my podcast some time ago, so you can go check that out and see what he has to say. This is just a summary of dual-use research and the NSABB charge. So they're basically saying here that research is good and we need research to proceed and have better life, but some of it can be dual-use, and some of it is inherent in a portion of life sciences research. However, the small subset that have the highest potential for yielding products that could be misapplied is called dual-use research of concern. And the NSABB defines that as anything that can be anticipated to provide products that would be used as a threat. So that's the charge of this committee. And they, of course, came into this story. They got these 2 papers from Fouchier and Kawaoka, and after reviewing them, they said, we can't publish this. We have to redact the papers. You know what redact means? You cross out parts in a black marker. So they were going to leave out data from these 2 papers and publish them in Science and Nature. So I wrote this blog post, and I called it a bad day for science, because the authors agreed to remove certain details from their manuscript, both the Fouchier and the Kawaoka manuscript. And so I wrote here, the article hints that details of the experiments may be made available to virologists with a legitimate interest in knowing them. So I ask, who will decide what constitutes a legitimate interest? And what if a virologist or another scientist who does not work on flu has an idea and would like the details? Will they be denied because they are not card-carrying influenza virologists? Science often works in unusual ways, and one of them is that difficult problems are often solved by individuals from different areas of research. So I find this abhorrent, the idea that you would publish a redacted form of a manuscript. This is not how science proceeds. You can't hide information. You always have to make it available. So the idea here, the NSABB said, these mutations allow you to create a pandemic strain. We can't publish them. But of course, we don't know if they allow the creation of a pandemic strain. But they could be useful. We don't know, but it could be useful. And if there's any possibility that they might be useful, they should be published. Now, the New York Times on January 2012 published an editorial on Sunday. This is very unusual that they weigh in on science on Sunday. Usually it's politics or economics or something else. And they called it an engineered doomsday. The New York Times called this virus an engineered doomsday. I guarantee you that the person or people who wrote this article never read the paper, didn't take a virology course in college, and knows very little about science. I'll bet someone called them up and said, you need to write this editorial and get all of Congress working on this. So here they talk about the work on H5. In the process, they created a virus that could kill tens or hundreds of millions of people if it escaped confinement or was stolen by terrorists. Any virus has the potential to kill a lot of people, so I don't see why this is any different. We always nearly champion unfettered research. In this case, it looks like the research should never have been done because potential harm is so catastrophic and the potential benefits from studying the virus so speculative. How can you make these conclusions if you didn't read the paper? Up to this point in time, I had not read the paper, but I didn't believe any of this. And I find it abhorrent that the New York Times chose to write this editorial without having done it. And you will see later how foolish they appear in hindsight. So I wrote a little editorial about this. I was really pissed off. I wrote it off in 10 minutes on Sunday morning, and you can go back and read it. Then the result, next thing that happened was there was a moratorium on transmission research on H5N1. This issue was getting too hot. So I think what happened on the face is that the authors of these two studies and other scientists got together and agreed for a 60-day moratorium on transmission research. And this is their statement here. No experiments with live or H5 reassortant viruses will be conducted, and they say we're halting this to give the community more time to explain the benefits of this research in an international forum where these issues can be debated. Okay, so this moratorium was supposed to last 60 days. It was just lifted a few months ago, so it lasted about a year. About 2 weeks after that New York Times editorial, they published a dialogue, a Sunday dialogue. I don't know if you know what this is, but in the week leading up to this, when it's published, they publish an editorial, and then you can write in comments, and everything is published the following Sunday. So I had written the Times a letter the week before, and so they invited me to participate in this. And this was the opinion piece that they put up. This is by Tom Inglesby, who is an infectious disease doctor and director of the Center for Biosecurity, University of Pittsburgh. The Center for Biosecurity. Universities now have academic departments on biosecurity. This has become big business, the whole business of protecting us against infections. Now, I don't debate that we should be prepared, but I think people who are in these departments, who depend on them for their livelihood, are somewhat conflicted in expressing their opinions about this. Now, this gentleman said here, second paragraph, ferrets are the best model for predicting human flu transmission. So the assumption should be that this new strain would spread among humans, perhaps as easily as seasonal flu. Ferrets are not a predictor of anything. Animal models do not predict anything. They are models. That's why we call them models. Let me give you an example. You work in a drug company. You're making an antiviral drug. You test it in rabbits. You test it in dogs. Do you then release it to people because the dogs and rabbits predict what happens in people? No, of course not. You do a clinical trial because animals are not predictors. That drug could fail in people because whatever happened in the animals was wrong. Same thing with transmission studies in ferrets. So saying prediction tells me he doesn't understand animal models. What else did he say that we should look at? He cites the 60% case fatality ratio. He says if this engineered strain were to escape the lab, and if it maintained high lethality, remember that, we're not sure it's lethal, while spreading as effectively, it could kill tens or hundreds of millions of people worldwide. So he says the potential benefits do not justify the potential dangers. I have a feeling he didn't read the paper either. He's not on the NSABB, and only those committee members could read this. So I responded to this, and they printed my response. Now, all this is going on in the US. And you know what the rest of the world thinks when we start telling them what to do? That's right. And the rest of the world doesn't like it when the US makes rules that everyone has to follow. So WHO had a meeting and they said, you know, this is really important research. We think it should be published. So this was a meeting where both Fouchier and Kawaoka presented, and WHO said they should be published. There's a preference from a public health perspective for a full disclosure of the information in these papers. And they said there are some concerns and we have to address these. We have to have a communication plan and a review of the biosafety and biosecurity aspect. I think that's fine. You put in place why this is important research and what are you going to do if the virus got out and you had to respond. Have a preparedness plan, as they call it in the business. So WHO said we should publish it. So I thought that was good because it meant it would probably happen. Then at a meeting in February in Washington, Fouchier gave a talk. And he showed the data, although he didn't show the mutations. And he showed how he took ferrets and he infected them with either the wild type H5N1 or the mutant virus. So he had 5 or 4 amino acid changes in this virus. He didn't tell us what they were at this time. That information is still classified by the NSABB. And he would show how the wild type H5 would not transmit by aerosol. So if you infected a donor and put it in a cage next to a recipient, the recipient wouldn't get infection. But his modified virus would transmit one time and a second time from this animal to the next. So transmits by aerosol. What about lethality? Remember, everyone's saying that this maintains its lethality. So let's look at that. This slide is a little messed up. These are actually screen captures from a video, a live stream of his presentation that I took at the time. And the lines are all messed up. But here we go. Intranasal inoculation, wild type H5, 2 out of 2 dead, 6 days after exposure. That's with 10 to the 6th virions. You do the same thing intranasal with his mutated virus, 1 out of 8. Only 1 out of 8 are dead. Okay? This virus is not highly lethal. How about by aerosol? Let's see. Wild type, and I'm sorry, these lines are way off, but here's aerosol wild type. So you infect a ferret and you put another one in a cage next to it, and you see if that ferret is infected. Of course, wild type doesn't transmit, so you can't look at the lethality. But we know the mutant transmits. So is that lethal? 0 out of 7 die. 0 out of 7. So why did everyone say this strain maintained its lethality? Well, that's because of this experiment, where he inoculated the virus intratracheally. You put a tube down into the trachea and you pour virus in. And then either the wild type or the mutant, which is off the slide here, kills everybody. You have to put it intratracheally. If you transmit by aerosol, it doesn't kill anybody. So Fouchier apparently said to that reporter the first time, it maintains its lethality, but he forgot one important fact. It maintains its lethality by intratracheal inoculation. That's why when I taught you about virulence, I told you the route of inoculation is really important. So see, this was all a big misunderstanding. Paul Keim didn't know this wasn't by, this was by intratracheal inoculation, but it was in the manuscript, so I don't understand where the NSABB came away with this idea. So the NSABB, after Fouchier's talk, said, let's see that paper again. And they said, we have to look at it and read it and think about it, and then they ended up reversing their decision. After careful deliberation, the NSABB unanimously recommended that the revised manuscript should be communicated in full. The NSABB also recommended 12 to 6, the communication of the data methods and conclusions presented in the revised Fouchier manuscript. So they decided now, well, Fouchier and Kawaoka revised their papers a bit. I have both versions, and there's a lot of extra wording, but the data are the same. The intratracheal inoculation is there in both versions. So I don't know what the NSABB was looking at. You should go listen to TWIV when I interviewed one of these guys, and we talked about that a bit. And we also did a TWIV in Dublin last year with Ron Fouchier, who's right here, and he talked about it. And he said, I can't tell you the mutations, I can't tell you this, I can't tell you that, but we got into some interesting discussions, so check it out. Earlier last year, I was in a panel at the New York Academy of Sciences where we debated influenza and dual-use research. And they left me out of the picture. I was sitting here. This is Peter Palese, who I got my PhD with. This is an editor from Science. This is Michael Osterholm, who's director of the Center for Infectious Disease Research and Policy at the University of Minnesota. This is Arturo Casadeval from Einstein. And this is Laurie Garrett, who is a writer and on the Council of Foreign Relations. And these two were very outspoken against these experiments. And if you go look at this video, you will see the controversy that arose. I want to talk a little bit about Osterholm. He's been a vehement opponent of publishing these data. He wrote a letter on April 2012 to the NIH. So this is after the decision to publish had been made. He said he voted not to publish or to redact the Fouchier paper the second time. He said the agenda of the meeting was designed to produce the outcome, which is to publish the paper. He said there's been only a one-sided picture of risk benefits presented. There was no review to address reverse genetics in the wrong hands. So this is the phrase that I hate to use, reverse genetics, but he used it, so I'm duplicating it here. That's, of course, when you take a plasmid and you make mutations and recover virus from the plasmid. And he said, all these people are doing flu work, so they're all in conflict. And he said, many influenza researchers say influenza mutants can be easily synthesized in many labs. Some outspoken researchers have been underrepresenting the increased risk that would be entailed by full publication of the mutations versus the current situation where only the general outline of the ferret passage scheme is known. So he's saying we shouldn't publish these mutations because anyone could introduce them into an H5 virus and get it to transmit among ferrets and not be lethal. So what? I don't understand the point of this at all. I think, again, that he is conflicted because he's directing a policy institute whose job it is to find potential applications of bioterrorism, and he's not really looking clearly at this, but I could be wrong. Now, he said scientists can't police themselves. Bad things happen in laboratories. In 1977, for example, a new strain of influenza arose, a new pandemic strain. It turned out that it was very similar to strains circulating in 1950s. And this is thought to be some kind of contamination, possibly through a clinical trial. So he said this came from a Russian lab, but I don't know that that's ever been proven. He said we shouldn't approve the publication of the 1918 flu virus sequences, which we talked about. Remember, they were obtained from corpses frozen and used to reconstruct the virus. So he said we should have never done that. Of course, those have been published for years and nothing has happened. I don't think that it's a bioterror weapon at all. And basically, his approach is to be afraid of everything. We're afraid that people are going to get a hold of these sequences and make bad viruses. My view is what FDR said. The only thing we have to fear is fear itself. If you take science from a viewpoint of being afraid, you're never going to progress. And I think that's the climate we've moved into. We're afraid of doing scary work because we think bad people are going to use it against us. I think this is totally wrong, and we just have to keep doing science the way we always have. Then the US Senate got involved. They had a hearing where they brought all these players in, including Tony Fauci, who is the director of NIAID, Fouchier and Kawaoka, of course, started this controversy. People from the NSABB, including Paul Keim and Tom Ingleby, whose letter to the Times I showed you before. And all these people testified. Mike Osterholm was there as well. And Fauci basically said at the end, you know, it's important to do this work. He noted that a public misperception arising during the debates was that the aerosolized virus was lethal to the ferrets. That was not the case, he said. Can you believe this makes it to the US Senate on a misperception? I just find this incredible. But anyway, when the politicians start to get a hold of science, then you're in trouble. You don't want them regulating it. And so this was, I think, very bad that there was a hearing. Fortunately, nothing happened to it, but there are now rules in effect for doing dual-use research. These were finalized just a month or two ago in the US. So if you're working on any of these agents or toxins, anybody work on these, by the way? I certainly don't. Here's avian influenza virus, Ebola, foot and mouth disease virus, Marburg, the reconstructed 1918 virus, and a variety of others. And if you do any of these kinds of experiments, you enhance the pathogenicity, disrupt immunity, let's see, increase stability or transmissibility. There you go. There's the H5 experiment, alter the host range. If you do any of these things, You can do them, but you have to have a plan. You need to have a risk mitigation plan, which is approved by your institution and by the funding agency. So basically, if I want to work on H5 transmission, I'd have to have a plan for what to happen if the virus got out, what we're going to do, how we're going to respond to it, how we're going to treat people. Columbia would have to approve it, and then the NIH would have to approve it as well. So it's not straightforward, and it could take months and months to do this. And my understanding from many people in the field is that they can't be bothered with this, so they're going to just get out of H5 research entirely. So I understand the need to be careful, but I think this has an unfortunate effect. So how about the papers? What do they actually show? The science, it turns out, is really interesting. So here's the Kawaoka paper. What he did, he took the H5HA, just the gene itself, and he randomly mutagenized it, and then he selected for variants of the protein that could bind human sialic acid receptors. So he found 2 mutations, Q226L and 224K, which allow pretty good binding to alpha-2,6 and pretty bad binding to alpha-2,3. So it switched receptor specificity. Then he took this H5 and he put it into an H5N1 background. He made a reassortant with a 2009 H5N1 strain. All H5N1 except H5HA. And then he infected ferrets. He would recover viruses and sequence them. He found 2 more changes in viruses recovered from ferrets right here and here. So he built all 4 of these changes together, 1, 2, 3, 4. And he finds that those 4 allow the viruses to transmit by aerosol from ferret to ferret. There's no mortality, just like the Fouchier experiment. And here are the mutations. Interestingly, one is way down here in the stalk of the HA. Here's the sialic acid receptor binding site at the top, and there are 1, 2, 3 proximal mutations, which probably affect sialic acid recognition. This one is very interesting down here. This apparently affects the stability of the protein. Now, so what we think, or what Kawaoka thinks, is when he selected for these mutations that allow this HA to bind human receptors, it destabilized the protein. So you needed this compensating mutation to stabilize it so it would be transmitted by aerosol. This was a totally unpredictable, unprecedented finding. And this is the kind of stuff that justifies doing these sorts of experiments. And I guarantee you that the editorial writer of the New York Times had no clue about this, and even if he or she had read the paper, they wouldn't have understood it. And this is one of the reasons I teach this course, because someday if you read stuff like this, you'll understand it. And if you don't, you should email me, okay? This virus, by the way, can be controlled by current vaccines and antivirals. So this transmissible H5H1 you can prevent it infecting by those means. The Fouchier paper did a different approach. He took H5N1 virus. He introduced a couple of mutations here, Q222, G224 in the black asterisks. He introduced those to allow alpha-2,6 sialic acid recognition. Then he introduced a single amino acid change into PB2 protein. This is a subunit of the RNA polymerase of the virus. You need this change to allow the avian RNA polymerase to replicate at mammalian temperatures. Okay, remember that. This is really important. Kawaoka didn't have to do that because he had a human virus with the H5HA on it. Then what he did was he infected ferrets. And remember, it didn't transmit by aerosol with just these 3 changes. So he passed it from ferret to ferret. And in the end, he got a strain that transmitted by aerosol among ferrets. And those viruses had these 2 extra changes, T156A and H103Y. So those are the 4. 1, 2, 3, 4. You know what they are. It's published. Everyone knows what they are. I don't think it does anyone any good in a bioterror sense because these viruses attenuated in ferrets. All right, so that's the Kawaoka experiment. Just a couple of weeks ago, a paper came out where they solved the structure of the Kawaoka H5HA in complex with human and avian sialic acid receptors. So here's the 3-dimensional structure of the H5HA. It's the Kawaoka adapted H5HA. And here's it in complex with the human receptor. And here it's complex with an avian receptor. Now remember, this HA really prefers binding the human receptor. It has like a 200-fold greater affinity for the human than the avian. But the avian receptor will bind well enough to get a crystal structure. even if you don't appreciate the details of this, I think you can see that the two sialic acids are sitting in the receptor pocket totally differently. Look at the red part up here in the human receptor. It's to this part of the sialic acid binding area, and here it's on the left. It turns out that, and remember, this is an H5HA binding a human sialic acid. It turns out that the H2 and the H3 pandemic viruses that infected people in 1957 and 1968, these all originated from birds, and they switched sialic acid specificity from alpha-2,3 to alpha-2,6, and they bind sialic acid just like this. So it looks like this is on the way to being a human, an HA, an H5HA that could infect human cells, at least just by receptor recognition. We know there are other things you also need to infect and transmit. But this is quite interesting in that the sialic acid, the human sialic acid, is fitting in the same way as it does in human strains. So now we can go out and look in nature. We know that there are H5s that are beginning to trend towards alpha-2,6 receptor adaptation. The bird guts have alpha-2,6 sialic acid, and for reasons that are not clear to us, these H5 strains are slowly adapting to using alpha-2,6. And so if we keep an eye on the structures, we can possibly predict when those are going to be a threat. So another reason why this research was so good, and you couldn't have predicted any of this. All right, now if you did a Google this morning or yesterday, you would come up with these two articles. If you just, I think I searched for H5N1, that's all, and these two were the top hits under news. May 4th, Chinese scientists create new killer flu. And that's from the Independent. Here's one, I forgot where this is from. Anyway, made in China killer flu virus. So this has just started again. These went away for a while, but these refer to an experiment which was just published last week, where this question of H5N1 transmission was addressed. But these are both wrong. I mean, a new killer flu wasn't created. And in fact, there are quotes from virologists in these articles. Immunologists expressed concern Friday about the dangerous work of scientists in China who created a hybrid flu virus that can spread in the air between guinea pigs. We've gone from ferrets to guinea pigs. And here another one. Senior scientists have criticized the appalling irresponsibility of researchers in China who have deliberately created new strains of influenza virus in a veterinary laboratory. So I would contend that these statements are wrong. They are not, it is not appalling irresponsibility. These are really good experiments. So I want to tell you what they are before we finish up. So this is the paper published in Science. There's the link. H5N1 hybrid viruses bearing 2009 H1N1 virus genes transmit in guinea pigs by a respiratory droplet. Now, so the guinea pig is the other model for transmission. It's a little cheaper. It's not as good as the ferret, and it tells you different things. But what they did here is the question, can H5 reassort with human viruses? So 2009 H1N1 viruses are still circulating in people and in animals. Could these two viruses reassort? And then if they could they transmit among mammals, and in particular among guinea pigs? So they made all 127 different reassortants of H1N1 and H5N1. They took the plasmids encoding each gene and they mixed them in different ways and got viruses with all possible combinations of these segments. It's an amazing amount of work here. And then they would test them for transmission in guinea pigs. So I made a figure to summarize all this. And this is broken down into highly efficient transmission, efficient, less efficient, and then no transmission at all. So the H5N1 virus they start with, it's all the segments are in red. This does not transmit among guinea pigs. The pandemic H1N1 transmits very well. And now if you start mixing and matching, all of these transmitted highly efficiently. So look at this one. This is quite interesting. This is H5N1 with just a polymerase subunit, PA, from the H1N1. That transmits very efficiently. Another one with just the non-structural gene transmits very efficiently. Remember, all of these have the HA, the H5HA that recognizes alpha-2,3 sialic acids. So I'm not sure how important these are, because it turns out that the upper tract of guinea pigs are different from ours and that they have alpha-2,3 sialic acids there. So this might not be a relevant experiment. Anyway, it's interesting that you can make this virus quite transmissible with 2 internal genes only. This one is efficiently transmitted. This is the H5N1 with just the HA from the H5 strain. So this is not as well transmitted as the others, but it can be transmitted. Presumably there's some incompatibility that is making it less efficient. These are not transmitted very well at all. So it's a very interesting experiment, I think. I don't think there's any danger in doing this. I think it addresses an important question. I have no fear that if any of these got out, they would cause a human pandemic. These are, in fact, none of these are lethal in guinea pigs. These are just being transmitted. So I encourage you to not believe these headlines that you're going to be seeing for the next couple of weeks, because I don't think they understand the science. So here's my view, as if you didn't know it already. I think nothing that a well-intentioned scientist does is too dangerous to publish. I think if you restrict publication, you get rid of serendipity, which is one of the main drivers of science. Think about it. We're going to restrict publication. That impedes us. It doesn't really impede the terrorists because they can get the information anyway. And what's happened in the last 2 years with H5N1 is people have gotten afraid. We're afraid, because of bombs that cause terror, and we think that infectious agents could do the same thing. But that really is restricting our progress, and I think we have to get away from that. Now, I have no problem talking about biosafety. I think being prepared is great, but let us do the research that needs to be done, and let's stop with the doomsday headlines. Now, I've written, and I think another aspect of this story is the importance of communication. As I've told you, the transmission saga was likely a result of miscommunication, or should I say a failure to communicate, from the famous Steve McQueen line, right? Paul Newman, thank you. Oh, a moviegoer, excellent. Was that Silverstein? Yeah. You would be the only one who would know. Are you sure? Positive. Okay. Most of the individuals who were against doing the workers' publication had not read the paper. If they had, they didn't understand the implications. So another reason why I want to teach you and everyone else virology. I think it's really important that we stop misunderstanding what's going on here. So I wrote an article in MBio last year about that. It's called Science Should Be in the Public Domain. So if you want to know my thoughts about that, you can read it. Let's end up with the latest avian influenza virus, which is infecting people, and that is H5N1, which emerged in China in March of this year. And this is the first time this virus has been shown to infect people. There have so far been 127 infections with 27 deaths, so the case fatality rate is 21%. But again, we don't know how many infections there actually are, so this may be artificially high in terms of lethality. In China, and it's been found in several places in China, they have isolated almost identical viruses from chickens and pigeons. So this is thought to be a poultry-associated infection, but the exact source isn't known yet. They're swabbing tens of thousands of birds in China at the moment and looking for more evidence of this virus. But at the moment, we don't know where the virus actually came from. And importantly, we don't have human-to-human transmission yet. I shouldn't say yet. We don't have human-to-human transmission. I personally don't think we will see it because this virus has evolved to be as it is now, and I don't see why it should acquire transmission. But if it does, then we can sequence it and find out what controls transmission of this particular virus in people. So that would be an interesting opportunity. So this is the geographic location of the cases in China, all on the East Coast here, as you can see, and you got the cases and the number of deaths. Began in Shanghai, but you can see a large number of cases in neighboring provinces as well. This virus turns out to be a triple reassortant. So there's a very large database of avian influenza virus sequences. So when these strains, here's one of them, avian origin H5N1, when it was sequenced, you could immediately search the database and say, oh, what's it closest to? So it looks like it has gotten genes from a duck virus, an H793 duck virus, a virus isolated from a brambling, an H9N2, and some H5N1 virus from wild birds. So you can see the segments come from each of these viruses most likely. Where this reassortant occurred is not known, and where the final infection to humans took place is still not known. So this is an ongoing story. The sequences tell us a couple of interesting, yes?
What was that gap in one of the segments?
That gap, it's an artistic mistake, I think. Yeah, that doesn't, it's not meant to mean anything. So the sequences reveal there's no multiple basic amino acids at the HA cleavage site, so this is not going to be a high pathogenicity virus like H5N1. It has the Q226L amino acid change, which is one of the ones put in by Fouchier, which allows binding to alpha-2,6-sialic acids. So this is probably adapted to human receptors. And look at this, it's got the PB2E627K mutation. That's the one that Fouchier introduced to get the H5 to replicate better at the high temperatures of mammals. Remember, avian viruses, that birds are cooler than mammals. The viruses aren't well adapted to the higher temperatures. So it's very interesting that that's in there. The other thing I want to tell you is that in that 2003 outbreak of H5N1 in the Netherlands, so 89 human cases, 1 death. The person who died had a virus with this change in it. None of the other 88 people who were confirmed infected, none of them died, none of them had this change. So this may be important for transmission, for lethality in humans anyway. We don't know anything about transmission. So that brings us to the end of virology, and here we are at the beginning of the course where we're wearing cold coats and scarves, and it's very cold, and before you know it, the course is over. I cannot believe how quickly it went. Just a couple of things before you leave. There is a survey at Courseworks. Please fill it out so that we know what to fix next year in the course and what you like. Finish the quizzes, because they're 10% of your grade. You can still do them. I'm going to have office hours Friday. I will announce exactly when in a day or two. Some people want to take the final on another day because they have 16 finals on Monday. That's fine. We're trying to find a day for that, so keep in touch. And I want to thank you for taking this course. I truly enjoy, and I speak for Dr. Silverstein as well, I truly enjoy teaching you virology. You can see this is an important life science for you to know. And the last thing is I want you not to forget what you learned here. So thank you very much.